Okay, hello everyone. Let's just wait for the students to join, then we'll start off with our class. Good evening, Somnath. Let's wait for the students to join in, then we'll start off with our new second class of the free marathon series. And today we'll be discussing about the restriction digestion, again one of the concept in unit number 13, that is methods in biology. Okay, let's wait for the students to join in first, then we'll start. Is the audio and video clear to everyone? Just let me know, confirm it please. Good evening Madhu. Good evening Mahesh. Good evening Vishali. Let's wait for students. Students are still joining in. Let's wait for them. Then we'll start the class. I hope yesterday the class was helpful for you all, and if you are new to Botany Insider, new to this particular series, make sure that you do not forget to hit the subscribe button as well. Good evening Niharika. Good evening Parvati. Hello Harshit. Good evening Sonam. So today also first we'll be completely understanding the concept of restriction digestion, then we'll be solving a previous year question as well. So stick to the video till the end of this complete lecture. Also download our application from the iOS, also from the Android application if you haven't yet downloaded. Make sure you join us there as well. In addition, these are the various things, various contents available for you. We have a theory session, we have general aptitude course going on, we have notes booklet, PYQ pointers, we have in addition to that various other courses which are going to be launched very soon, so join in there as well. Good evening Sambhavi. Okay, so the new course starts on 11th. It would be launched very soon, so if you are targeting the upcoming exam, join in that course. It would be launched on Friday on our application, on our YouTube channel as well. So make sure that you, if you feel like that these classes are helping you out, join us in that particular course. It would be a 60-hour course in which we'll be covering all the concepts of CSIR UGC NET Lifescience stream. It would help you to revise the concept better. So talking about today's class, let's start off with the basics. Very first thing, what do you mean by restriction? Like we have the topic that we are going to discuss is restriction digestion, right? So what do you mean by restriction at the first go? Because this is the first term, this is the first content that we have to look forward to. So what is restriction? Restriction का simple मतलब क्या होता है? It means to stop something, or we can also call it as किसी चीज को रोकना, right? And if you look into the word of digestion, digestion could simply be called as to break down a bigger thing into smaller particles, right? And agar if we apply this into our concept or into our main procedures of the molecular biology, what is it actually? In this, what we are doing, we are trying to prepare the DNA for the analysis. Because see, very simple logic, just understand it this way. If you want to, let's say this is the DNA that you have, right? Double helical DNA that you have. Now if you want to analyze this DNA, can we analyze the complete DNA and at the first go? Or in other simple words, let's say that if you want to qualify CSIR, can you qualify the CSIR reading all the 13 units all together like in a single day? The answer would be no. We have to divide the content into various parts. We need to divide the concepts into various small chunks, right? Same happens with the DNA analysis as well. If you want to analyze the DNA, we need to break it down. We need to have small chunks of it. We need to have small parts of it, so that जो उसका analysis है, जो हमें part को, the part which we have to actually analyze, it becomes easy for us, so that First, obviously, if you have a smaller chunk, then you can take the first chunk, analyze it, go to the second chunk, then analyze it, and so on and so forth in a systematic manner. You will be able to do the work. But on the other hand, if let's say the restriction digestion is not there, so what will happen? This complete analysis, like if you want to analyze this complete DNA, it would become a bit difficult. So that's the reason why the restriction digestion is of utmost importance. So now let's look into the words. A restriction digest, it is a procedure that is used in molecular biology. Why it is actually used? In order to prepare the DNA for what? For the analysis and other processes. Second thing, it is sometimes also called as DNA fragmentation, because we are making the fragments of the DNA, right? So we can also call the restriction digestion as DNA fragmentation as well. Now, if you want to give it a technical term, if you want to give it a technical definition, so what is restriction digestion all about? So it is a process of cutting the DNA molecules into smaller pieces with special enzymes, because obviously we are cutting a DNA. It is not a simple thing. You want to cut a DNA fragment, like the complete DNA need to be cut down. So for that, for this restriction digestion, we would require some special type of enzymes, and these enzymes are given the name of what? They are given the name of restriction endonuclease, or we can also call them as restriction digestion enzymes. So, and abbreviated as, they are abbreviated as what? They are abbreviated as RE, restriction endonuclease. Now let's understand about what does endonuclease mean. See, restriction endonuclease, what does this mean? See, there are two types of nucleases. There are two types of main contents. The first one is endonuclease, and we also have exonuclease. Okay, but we are taking restriction endonuclease for the cleaving of the DNA. Why? Because there is a specific reason to that, and that is, let's consider this is the DNA that you have to cut down, and the endonuclease is the one which would be cutting the ends in the inner side. Endo means in, exo means ends. Okay, so exonuclease works on the ends, endonuclease works in between. So this concept is majorly in unit number 3, that is fundamental processes. So make sure, if you want to like cover up in greater depth of this content, you have our courses, so join in there. But for the time being, just understand one thing, and that is restriction endonuclease. The endonucleases are the one that cuts at the inner side, like on the inner side of it. So this is what restriction endonuclease is all about. And in the case of restriction digestion, we use restriction endonucleases enzymes. Is this clear to everyone till now? If yes, let me know. Is it done? Understood everyone? Can we move forward? Yes. Is everything clear till now? Very good. So, this was about it. Let's move forward now. Now let's understand about some more terms. Restriction digestion. Restriction digestion is also called as restriction endonucleases enzyme. Okay, it is being used to cut the or digest the DNAs at specific sites that is dictated by the surrounding nucleases. Now first my question to you is, try to define endonucleases according to the understanding that I have just given you. Try to define what an endonuclease is all about. Yes, what is an endonuclease? Quickly. What is an endonuclease according to the content I have just explained you? Am I audible? Hello, hello, hello. Yes, the DNA that comes from or that cuts from the inside. Yes, very good. So it is being used, this restriction endonuclease, it is used to cut or digest the DNA at specific sites, because obviously if we are analyzing, if we are observing, if we are actually cutting the DNA, so there need to be a specific site as well, right? So this particular DNA, what it would be actually doing, this restriction endonuclease, it would be cutting the DNA at a specific site. Okay, so what are these specific sites? So these specific sites, they are known as the recognition site. See, all these terms are very, very important, be it restriction digestion, endonucleases, specific sites called as the recognition sites. Remember about these terms, because all of these are technical words, all these are technical terms which you need to understand. Okay, a restriction digest, it is a procedure that is used in the molecular biology to prepare the DNA for its analysis or other processing. So whatever we have discussed till now, this is one line for it to be like get summarized, right? So remember about it. Restriction digestion, it is a procedure that is used in the molecular biology for what purpose? For preparing the DNA for its analysis and other processes. The DNA would be cut by the help of restriction endonuclease. It would be cut on the recognition sites. Okay, I hope everything is clear till now. Now in addition to that, we have other few terms, that is, they specifically cleave the nucleic acid at specific nucleotide sequences, which are called as the recognition site or the restriction site, which helps in the generation of what? For generation of smaller fragments. Clear? Now what it would be actually doing? So a restriction enzyme, it would be cleaving what? Because in the DNA, what do we have? We have sugar phosphate bond. So it would be cleaving the sugar phosphate bonds. So this is one of the additional thing that you can remember, that they are actually cutting down the sugar phosphate bonds. Now these restriction enzymes are also given the name of molecular scissors. Why? Because they are helping to cut down, they are helping to break down or cleave the DNA into fragments. So first one we have is HIND2, HIND2. So it was the first restriction enzyme that was discovered accidentally. See, remember one thing, in scientific world, all the major things, all the most important experiments or the discoveries, they all happened like because of some sudden incidents. So even in the molecular scissors, even in the restriction enzymes, the first one, it got discovered accidentally. So HIND2 recognizes what? It recognizes and cut the DNA at these two sequences. So these two are the recognition sites for the HIND2 molecular scissor. Okay, now you have this complete list. In this list you have a lot of enzymes given in front of you. from where they are actually found. These are the recognition sites, and at these points they would be cutting. There is nothing to explain in this, you have to remember about it. If not the recognition site, but if you are preparing for GATE and for the DBT exams, this thing is again very very important. In addition, remember the source as well. You can take a screenshot of this from the class itself and you can remember about this. You can remember about all the sources, all the recognition sequences, all the areas where it would be actually cutting. So this is a list for you all. Make sure you remember this, do not miss it out. Now we have the types of restriction digestion. Okay, now we majorly have 3 types of restriction digestion enzymes. First one is restriction digestion 1, then we have type 2, type 3. So what are these? We have type 1, type 2 and type 3. Let's understand about each one of them. Before that, if you have any doubt, any difficulty, let me know, then we'll start off. Very good, Laiba. Everyone, is it clear? Just let me know, then we'll start off with the classification as well. Very good. So now let's understand about the types of restriction digestion. Okay, restriction digestion enzymes. So first one is the type 1. We have 3 types, type 1, type 2 and type 3. So what is the type 1 all about? So type 1 enzymes, they are the ones that exhibit both the restriction digestion and the DNA modification activities. Now every single point written over here is very very important. You can get it in both ways, in the statement type question as well, and in the case of the practical analysis, like the one which we are going to do after the class, like at the end of the class. So in both ways you can get it. So please remember about each of the topic, each of the point given to you. So they require what? They require the cofactors. Remember about it, that type 1 restriction enzymes require the cofactors. For example, magnesium 2+ and SAM, that is S-adenosyl methionine, and the ATP for their activity. Now over here you can correlate it to a specific plant hormone which uses S-adenosyl methionine for its biosynthesis. I won't be naming it here, but if you have worked it, if you have worked properly, you will be able to correlate, because interlinking of units is very very important. Remember about it. Now type 1 restriction endonuclease, it cleaves what? It cleaves the DNA at specific or at non-specific sites, and that can add up to 100 base pairs and more from the recognition sequence. So what is it actually doing? What is the type 1 restriction endonuclease actually doing? So it cleaves the DNA at non-specific sites that can add what? That can add up to 100 base pairs or more from the recognition sequence. Now because of these features, type 1 are less used. Now why they would be less used? Because see, we are doing the complete process, we are doing the complete concept for the gene work, and in gene work, usage of a chemical, usage of a enzyme for the non-specific site is not possible, right? So type 1 restriction endonuclease is lesser, or it is in the lesser use for the gene manipulation. Then we have type 2. So type 2 is the one that is most commonly used. It is the one that is like majorly used for all the gene cloning processes. The type 2 enzymes and their corresponding modifications, methyl esterase, acts as separate proteins. Now what is the function, or what are the main advantages of type 2 in comparison to 1 and 3? First is that the restriction modification are mediated by separate enzymes, so it is possible to cleave the DNA in the absence of the modification as well. Second is the restriction activities do not require cofactors, as we have just studied that in case of type 1 what was there, it was requiring cofactors, so it becomes more like energy dependent condition for the cell to work, right? So in this case what is happening, DNA modification activities, these are the activities. See, the DNA is initially in a nascent form where it is not having any kind of modification, but if it wants to do any specific work, we need to like attach it, we need to have some kind of modification done in it. For example, methylation, for example adding sulfur to it, because when you study about translation, in translation concept, after that we have various DNA modification contents where we change the DNA, we change the complete sequence of the DNA. Yes, methylation, acetylation, yes, very good. So there are in addition to that there are a lot of DNA modifications as well. Histone modification, yes. So these are all the DNA modification activities that are done after translation in order so that the plant, the genome could be worked in a better way, and the final result, the final work that have to be done could be done in a more enhanced manner. Okay, so this was about it. I hope your doubt is clear. Now the restriction activities do not require the cofactors, for example the ATP or S-adenosyl methionine, making them easier to use. So because obviously, see, what is it written? In case of type 1, what we are using? In type 1 there is usage of cofactors, but in type 2 there is no usage of the cofactor, so it is easier for the cell to use it. Last point, these enzymes are site specific, which is the very first requirement for the accuracy of gene manipulation, right? So for gene manipulation, site specificity is required, because there need to be a specificity, there need to be a cross checking done so that the gene could not get any hampered, the gene does not have any kind of hampering done. So the genes are this type 2 enzyme is site specific, as they hydrolyze the specific phosphodiester bonds in both the DNA strands. Third, we have the type 3. So the type 3 enzymes are the one, they are similar to type 1, but there's a bit difference, that is they recognize the specific sequences and they cleave 25 to 27 base pairs outside of their recognition site in the 3' sequence. That means when you look into the DNA structure, we have two strands. First is in the 5' to 3' direction, second is 3' to 5' direction. How are these formed? We need to go into depth of fundamental, so we won't be able to go in depth at this point. But what is it saying? That the type 3, it require or it recognizes specific sequences, and they cleave at what? They cleave at 25 to 27 base pairs outside the recognition site. For example, let's say the recognition site was here, so it would be cleaving 25 base pairs outside that thing in the 3' direction. Again, it is a bad thing for what? For the gene manipulation process, right? So that is the reason why type 3 is also not used. In addition, they require magnesium 2+ ions for their activities. I hope till here everything is clear to everyone. If there's any doubt, any difficulty, you can ask me, then we will be moving forward. Clear everyone? Very good. Let's move forward now. Now let's understand about the restriction digestion. See, the DNA is of two formats. let's just understand it. Just a second. Yeah, see, the DNA could be either linear, right, or it could be circular. In any way it could be present. So depending upon the same thing, let's just understand the number of fragments that are formed in it. So let's say first it is in the linear case. So you'll solve it yourself, you'll get the concept very easily. Okay, now if it is a linear fragment and you are given that there occurs restriction digestion at site 1, site 2 and site 3. So how many fragments would be formed? Just try to analyze and give the answers in the comment sections. Yes, you have this DNA fragment. So how many fragments get formed when there occurs digestion at 3 points? Try to analyze. Very simple, just try to analyze and give the answer. Yes. Quickly. Mega, let's discuss about it at the end of the class. First let's complete it, okay, then we'll discuss about this thing as well. So now write down the answer first. 4. Okay, very good, Madhu. Others, what's the answer? Try to solve it. Tejasvi is saying 3. Tejasvi, try to correlate it. Okay, Laiba is saying 4. 4, very good. Tejasvi, try to analyze it again. Just calculate. See, there are 3 areas where the cutting is getting done. Cut is getting done. So we have 1, 2, 3 and 4 fragments. So what does it mean? That there would be 4 fragments formed. Remember about it. So over here we have our formula that the number of restriction digestion site when it is n. So the fragments formed in case of the linear DNA would always be n plus 1. Over here we had 1, 2 and 3 restriction sites. So n was equal to 3. So fragment would be equal to 3 plus 1, 4. Okay. Remember about it very, very nicely. So I hope Tejasvi and Vaishali, your doubt is clear now. Why is it 4 and not 3? You just have to count, nothing else. Just see where the cut is getting done and then just count the number of fragments. And now just try to get the answer for the circular DNA. You have this circular DNA and over here also we are having 3 fragments, or sorry, we are having 3 cuts or 3 restriction sites. So how many fragments would be formed in case of the second case? Very good, Vaishali. Analyze the second one and get the answer for second one now. Yes, in the second one. Yes, Madhu in second. Let other students also do it first. Very good, very good. Sambhavi, try to analyze for the circular. Again, just count. 3 sites are there. First fragment, second fragment and third fragment. So we'll be having 3 fragments. So for the circular case, the formula is that the number of restriction site, if it is equal to n, so fragments would always be equal to n only, okay, because there is no end to it, right? If there is no end, just count and you'll get. So in case of linear, 1, 2, 3, 4 plus 1 would be there. In case of linear, we would be having 1, 2 and 3. Yes, in linear plus 1, in circular, total equal to the same one. Very good. I hope this concept is clear. Same is written over here. See, and in this as well, we have partial and complete content, like partial and complete concept is there. So when it is complete digestion, number of fragments would be n plus 1 in the linear, and in case of partial digestion we would be having number of fragments equal to n, because what do we mean by partiality? When the work is not getting completed up till the end point. So in case of linear DNA, partial digestion, then the number of fragments would equal to n, which is equal to restriction sites. Complete DNA or complete digestion, n plus 1. So again important concept. Then in case of circular DNA में what will happen? Partial digestion, number of fragments will be equal to number of restriction sites, but अगर हमें partial देखना होगा, so उसके अंदर it would be according to the question, like whatever is the question given to you, the conditions given to you, in that particular condition we will be having our answer solved, especially in case of circular DNA. Use this presentation in the best manner possible. We have tried to add every single small bit to it, so do not skip out even a single slide. Okay, make sure you understand, make sure you remember about it in the best manner possible. I hope this concept is clear. See, what does partial and complete digestion mean? Partial means which involves the performing of incomplete digestion of the plasmid DNA. Complete means when the complete digestion is there. And there is one more thing, that is double digestion. What is double digestion? So when two restriction digestions are used to digest the DNA in the single reaction. Simple. Double digestion means when you are using two restriction digestion enzymes in a same reaction. Okay, and ओके यू वांट टू ओके ठीक है. देखो डबल डाइजेशन क्या होता है, व्हाट इज डबल डाइजेशन? जिसके अंदर, जिस कंप्लीट रिएक्शन के अंदर वी आर यूजिंग टू रिस्ट्रिक्शन एंजाइम्स, जहाँ पर हम दो रिस्ट्रिक्शन एंजाइम्स को यूज़ करेंगे. किस चीज़ के लिए? हमारे डीएनए को डाइजेस्ट करने के लिए. सिंगल डाइजेशन या नॉर्मल डाइजेशन में वी आर यूजिंग ओनली वन रिस्ट्रिक्शन एंजाइम. इन केस ऑफ डबल डाइजेशन हम क्या करेंगे? हम दो डाइजेशन एंजाइम्स को यूज करेंगे इन ऑर्डर टू गेट आवर वर्क डन. ओके, सो व्हाट आर द फैक्टर्स अफेक्टिंग? कौन से फैक्टर्स अफेक्ट करते हैं हमारे रिस्ट्रिक्शन डाइजेशन एंजाइम की? सी, फर्स्ट थिंग इज टेम्परेचर. अगर किसी एंजाइम को वर्क करना है, मोस्ट इंपॉर्टेंट चीज़ क्या है? द टेम्परेचर शुड बी ऑप्टिमम. सो टेम्परेचर शुड बी ऑफ द एटमोस्ट रेंज जिसके अंदर एंजाइम वर्क कर सके. देन वी हैव द बफर सिस्टम, ionic condition and the methylation. All these 4 points are very very common, because see, अगर एक enzyme को, if any enzyme have to work in a proper manner, what is required? The temperature should be optimum, the buffer system should be in the pH range of 7 to 8 so that the work could be done properly, and from here you can get statement wise question. So make sure you remember about these things as well. Then we have ionic condition and last is the methylation of the DNA. That means the methylation of the specific adenine or cytidine residues within the recognition sequence that affects the digestion of DNA. Okay, so these are the factors. Now we have the main thing as the applications. The other 3 points are not of that use. We'll understand it some other time. So what are the applications of restriction digestion? So these are the application. It is used to map the DNA molecules. It is used to analyze the population polymorphism. It is helpful in rearranging of the DNA molecules. Then we have the application of preparing the molecular probes and to create the mutants as well. So these are the main important applications of the restriction digestion. So this was the main concept about the complete topic. Now we have a question. Let's quickly go through the question and understand the main concept behind it, like how to solve. It is a previous year question. Let's understand the previous year question, try to solve it, then we'll do it together. Okay, read the question, you will get it, try to find the answer, then we'll solve it together. It's a very simple question. If you put in a bit of logic to it, you will get the answer for it. Okay, let's just read the question carefully. The question says, a researcher attempted to clone a 630 base pairs coding sequence for the gene downstream to a bacterial promoter, which is 500 base pairs in length. Okay, so we have the promoter of 500 base pairs, we have a gene which is of 630 base pairs for the overexpression and the purification of the encoded protein. The gene sequence was isolated as a small fragment. Over here we have the recognition sequence. Arrow indicates the site at which the restriction is getting done. Now what you have to do, there was a double digestion done. So what is double digestion? We have just understood about all of these terms in the conceptual understanding. Double digestion is when there are two enzymes getting used. Which are these? First we have HIN3, then we have EcoR1. So question is, which one of the following restriction pattern? क्वेश्चन को जूम कर दीजिए. जूम कैसे किया जाए? रुकिए बच्चे, लेट मी ट्राई आउट, गिव मी अ सेकेंड, लेट मी ट्राई टू जूम इट फॉर यू ऑल. आई होप क्वेश्चन अब विजिबल होगा सबको. ओके मेघा, आई गेस क्वेश्चन अब आपको विजिबल होगा. रीड द क्वेश्चन क्विकली, देन वील सॉल्व इट एंड देन वील ट्राई टू अंडरस्टैंड इट. Is it clearly visible now? Okay, get the answer for this question now. Then we'll quickly solve it. It's a very simple question. Give it a try, you'll get it. Done? Quickly write the answers in the chat section. Option A, very good, excellent. Madhu have got the concept. Other students get the answer very easy. जो अगर आपने क्लास में अच्छे से सुना होगा, so it would help me out. आप आराम से इसको सॉल्व आउट कर पाएंगे. Yes, Madhu, the correct answer for this question is option A. That means आपको पूरा concept अच्छे से समझ आ गया. That's very good. So you utilized your 40 minutes in this session और एक बहुत इंपॉर्टेंट कॉन्सेप्ट लाइक दिस रिस्ट्रिक्शन डाइजेशन कॉन्सेप्ट इज वेरी वेरी इंपॉर्टेंट एंड इट्स अ बिट ट्रिकी एज वेल, सो इट्स वेरी गुड दैट यू गॉट द कॉन्सेप्ट. सो now let's just try to solve it because otherwise it will take a lot of time. Just look at the question. See, the promoter is of 500 base pair length, yes. The total is 630 base pairs, so the gene is what? The gene is 630 base pairs and the promoter that we have is 500 base pair in length, right? And the restriction is occurring at SAM1 where it is present over here. Now the gene is present where? It is present at this particular position, like in the 630 base pairs upstream की जगह पर हमारा eco R1 30 base pairs in the upstream present है. डबल डाइजेशन हो रहा है हिंट 3 और इको R1 से. जब हमने इंसर्ट को फाइनल किया, सो इंसर्ट को फाइनल करने के बाद व्हाट वील बी हैविंग, यहाँ हमारा हिंट 3 प्रेजेंट है, वील बी हैविंग आ प्रमोटर. प्रमोटर की लेंथ इज 500 बेस पेयर. वी आर जस्ट राइटिंग डाउन एवरीथिंग जो हमें पेपर के अंदर जो हमें क्वेश्चन में गिवन है. देन 30 बेस पेयर्स के बाद इको R1 साइट है एज अकॉर्डिंग टू द क्वेश्चन. देन जीन है, जीन के बाद यू कैन अगेन सी वी हैव इको R1, राइट. सो अगेन इको R1 इज देयर और हमारे पास दो डबल डाइजेशन के लिए है. वी हैव 2 एंजाइम्स फॉर डबल डाइजेशन विच इज हिंट 3 एंड इको R1. सो डबल डाइजेशन होने के बाद वील बी हैविंग some vector backbones. We'll be having one fragment of 530 base pair and the second fragment of 630 or 600 base pairs. So the answer would be option A, 530 base pairs plus 600 base pairs plus vector backbones. It's a very simple question. All you have to do is you have to be like very very analytical and you'll be able to get the answer. Very easy, and these type of questions are repeated in the exam as well. I hope this is clear to everyone. If there's any doubt you can go through the recording again, like this recording would be available to you over here itself. So अगर आपको कोई difficulty है you can understand it here. उसके बाद भी कोई difficulty आती है you can contact me. We have our application, download our application from the iOS Play Store both. It would be helpful for me because I am trying my best from my end to help you all out. All you can do is download our application, enroll in our courses. बहुत सारे courses available हैं, अभी और भी courses launch होने वाले हैं. We'll be having crash course, we'll be having general aptitude new courses as well because students are liking these courses. So make sure that you join in there. And if you are new to this marathon series, do not forget to like hit the subscribe button. Also share it to your friends, share it to your classmates because you all must be in masters or in the BSc. Somewhere around you must be having your friends doing the same preparation. Please share it to them. It would be helpful from your end to me. Thank you for watching this video. Tomorrow we'll be doing the third topic. I'll schedule a class for you all tomorrow as well. We are having our marathon for 7 days, 7 topics. All the topics would be very very important for you. Join in there and for sure it would be helpful. So thank you for joining in everyone. Let's meet tomorrow. Timing would be same 8 p.m. okay, so let's meet tomorrow now. Good luck and make sure you subscribe and you share the videos, share about the channel. We have to hit 10,000 subscribers very very soon. Okay, so thank you everyone. Let's meet tomorrow. Take care.